So, student, now I'm going to discuss Sanger sequencing. Sanger sequencing is also called chain termination method. History of Sanger method. This method developed by Frederick Sanger and colleagues in 1977. It was the most widely used sequencing method for approximately 25 years after its discovery. He got Nobel Prize in 1918. Introduction of Sanger method. Sanger method Sanger method is that method to find the nucleotide sequence of unknown DNA strand. We can find the nucleotide sequence of that DNA which are unknown DNA. More recently, Sanger sequencing has been upgraded as next generation sequencing methods, especially for the large scale genome analysis and for obtaining especially long DNA sequence reads greater than 500 nucleotides. So, what is the basic principle of Sanger method? The basic principle is generally is an in vitro synthesis of DNA strand. That is in vitro synthesis, and by using determinators dideoxynucleotide which contain dideoxynucleotide. The growing strand terminates at specific site. And secondly, upon termination, the strand or overlap to get original sequence of unknown DNA strand. As you can see, there are 3 to 5 sequence and their bases, and these strands are overlap and got original sequence of unknown DNA strand. DD-ATP in the reaction, anywhere there is RT in the template strand. Occasionally, DDA will be added to the growing strand. So, what is the requirement of Sanger method? Their basic requirements Sanger methods are single standard template, primer, DNA polymerase, dideoxynucleotide, the 3 hydroxyl group necessary for formation of phosphodiester bond is missing in DD and TPase. Every nucleotide has its specific DD-NTP from DD-ATP, DD-GTP. You can see at the diagram DNTP and DD-NTP. So that is the requirement of Sanger method. Let's move on procedure. There are 4 steps: denaturation, primer attachment and extension of bases, termination, and gel electrophoresis. The DNA template is treated with heat so that it becomes single strand, that is denaturation. A short single standard primer which is radioactively labeled is added to the end of DNA template. Add template DNA and primer in 4 tubes. Now add DD-NTPs in tubes in the way that single tube contain one type of DD-NTP. So extension is taut and bond formed of various sizes. The fragment of DNA are separated by electrophoresis. Overlap these sequences to find out sequence of target DNA. You can see in the diagram there are single standard DNA and these are the DDATP and these are DDTTP. These are the DDTTP and these are DDCTP and these are DDGTP. So they are grouped in the basis of their colors, and in electrophoresis we can find the basis on their basis of the colors. We can read it, so we can find the sequence, Dewey's DNA sequence. In electrophoresis we can use sequencing gel. that is slab or capillary gel. So we can read it and find the DNA sequence.